Good afternoon, Biology 12 students. This is Mrs. Becker again, and welcome to our second video. Today we're going to be focusing on the organelle functions. Last day we took a look at the animal cell. You guys had a chance to look at the differences between both prokaryotic and eukaryotic cells, and then you had an opportunity to draw and label the animal cell as well as the plant cell. Now we're going to take a look at each organelle individually and what they do to contribute to the functioning of the cells. We're going to start off with the idea of a nucleus, and this is a big cell right here. And this right here is the nucleus. Inside you see that dense part, the nucleolus, but this part right here, the nucleus, that is the largest organelle in a cell. It is a double-layered membrane, and that membrane we call the nuclear envelope. The nuclear envelope. So that's important to note. And because things are made here within this nucleus, it's really important that it does have holes in it. So there are nuclear pores, and those pores allow whatever material or substance is made within the nucleus to travel out of it, to get to the endoplasmic reticulum and move through the cell. It is the control center for the cell. It's often referred to as the brain. And inside, just like the inside of the cell contains cytoplasm, the inside of the nucleus contains nucleoplasm. Within that brain also is the chromatin. Now you might be used to the wording chromosome. Chromosome is what it's called when it's dense, just before it goes through cell division. When it's loose like a goose inside that nucleus, it's called the chromatin. The nucleolus is that dense part. It is within that nucleoplasm. Most cells only have one, but sometimes they will have two nucleolus within there. The nucleolus is the site of rRNA production, and this is really important and will come into play when we start looking at polypeptides and proteins and all that wonderful biochemistry stuff that happens inside the cell. In the nucleolus, that rRNA is made, and it starts putting together some proteins. So it does need to leave the nucleolus. It kind of happens inside there, but then eventually that will have to leave that structure. So that's why those nuclear pores are just so important in the functioning. Now the nucleus specifically directs that protein synthesis because it knows what needs to be made. It has that code. So the nucleus itself does the mRNA. And again, we're going to be going into tRNA and mRNA and rRNA in detail in about a week or so. But what does happen is that mRNA will travel into the cytoplasm and start combining with the ribosomes, and those ribosomes do have then the coding for the specific polypeptide or that specific protein that it is trying to make, that it's responsible or coded to make. So again, here's a bit of an idea of what it all looks like. You have your nucleus, you have your nucleolus. Here's the pores on that outside of the nucleus, and that allows substances to pass back and forth to get to this transport system, or ER. Here's some great electron microscope pictures of what that would look like. Now, we had mentioned the chromatin before, and the chromatin is the hereditary material of the cell. When it tightens up, when it condenses to form that well-known X, back in Science 9, when you did meiosis and mitosis, you would have seen those chromosomes. Well, when that's uncoiled, it's called chromatin, but it's still that hereditary material. When it's all coiled up, it has that beautiful X shape. It's a rod-like bodies in the nucleus, and it tightens up during that cell division. Here is an example of a eukaryotic cell. Now, we talked about this actually is an egg with a bunch of sperm trying to fertilize it. Now, we as humans, we have 46 chromosomes. An egg would have 23 and a sperm would have 23, so that when they fertilize together, you get that combination of 46. But not all organisms have 46. For example, a pea plant has only 14 chromosomes. A cat has 38 chromosomes. We as a human have 46. I find this quite interesting. A little difference between cat and dog-haired dog, 78 chromosomes. That's what its genetic code looks like, 78 chromosomes. Now, the cytoplasm is the jelly-like inside of the cell. It contains and supports all of the organelles, but it really is like a jelly. And around it is that cell membrane or plasma membrane. The cell membrane acts as a skin around the outside of the cell, and it's selectively permeable. So selectively means that it allows some things in and it keeps others out. It is a double membrane, and we're going to be taking a look at it in a little more detail later, but it's a double membrane, whereas most of the organelles are single membrane, so the vacuoles, the lysosomes. Some lysosomes can be double, but the ER, the Golgi apparatus, those are single membranes. Now, the double membranes, though, are mainly the nucleus, the mitochondria, and then that plasma membrane. Here's an example of what the plasma membrane or cell membrane looks like. You have your phospholipids, and in there you also have proteins. These proteins are very important and have a lot of role in some active transport. Now, here's the outside of the cell, this top part here. This is the outside of the cell, and you'll have your proteins, and you'll also have these carbohydrate chains. And this is what is used for cells to recognize self and non-self, so that it knows, hey, is that my cell, and is that what I'm supposed to attach to there? All right, now let's take a look at that smooth endoplasmic reticulum. Now, the smooth endoplasmic reticulum over here, it doesn't have ribosomes on it. It has a slightly different task. It still starts at the nuclear envelope and works out towards the cytoplasm, and it is that transport system of the cell. But because the smooth ER doesn't have the ribosomes on it, its main function is actually lipid production, lipid manufacturing. So for those cells that produce steroids, they typically have quite a lot of smooth ER. Now, there is a part of the endoplasmic reticulum that can bleb. Okay, and what that is it kind of, at the end, it pinches off, and then you have a vesicle, and that vesicle will transport something that needs to be transported, let's say, to the Golgi body, can then get boxed up and ready to be shipped off. So that transport vesicle is a very important part of the process of getting stuff ready to exit the cell. Now, there's also other enzymes in the smooth ER of the liver, and that is what helps us detoxify drugs and poisons. So those are really critical, and that is a big role of the smooth ER as well. Frequent exposure, though, leads to a real growth of this ER, and then you get an intolerance to the substance. So that can be really dangerous to the body as well. In the rough ER has ribosomes, and the ribosomes are the location of protein synthesis or polypeptide creation. So that is why rough ER, you're mainly shipping proteins. It will then often you'll have that blebbing of the transport vesicle that will take it right to the Golgi body, Golgi apparatus, Golgi complex, whichever one of those you use. One of the sides of the cisternae, so the Golgi complex, is made of these flattened cisternae or flattened sacs. And you have a receiving side and you have a shipping side. So because over here is where the ER might be, so let's say that's the rough ER, and then you have your transport vesicle that kind of pinches off, this would be your cis, or your receiving side. That's the side that things will come into to get ready. And then this side is your trans side, or your shipping side. And that is from there, it will ship things off to move to different locations, not just in the cell, but maybe it's shipping it off for exocytosis, which is exiting that cell. So many transport vesicles from the ER go to the Golgi body or Golgi apparatus. And the Golgi is the center of manufacturing. And it is especially extensive in cells that are specialized in secretion, right? It's getting everything ready to get out of that cell. Vesicles and vacuoles. Vacuoles are a lot larger. So here's an example of a vacuole. We did talk about this before in a plant cell. Here's that process that I was talking about before. You have your transport or your highway. A bleb comes off. Here's your transport vesicle. It then takes the substance or material or molecule to your Golgi complex, Golgi apparatus, which then packs it up and off it goes, more than likely to leave the cell. The lysosome is a membrane-bound organelle, and it is typically filled with hydraulic enzymes, which means that it's going to digest things. Truly, it is the ultimate recycler. It is going to break down anything into its parts. And often the parts get recycled. Not all the time. Sometimes they just get digested and then released into the cell. There's lots of special vacuoles formed by the Golgi body. That Golgi body is, in fact, a double membrane, as it was mentioned before. But lysosomes, they play a really critical part because they're going to get rid of anything that's kind of building up in the cell. And there's quite a lot. But sometimes those lysosomes, they don't work all that well. And there's a lysosomal metabolism problem. And there are some diseases, such as Pompei's disease and Tay-Sachs disease, that are due to non-functioning lysosomes. So Pompei's disease, that's when the heart and the skeletal muscles, the lysosome in them doesn't break down the glycogen, and you get a buildup of glycogen, which then causes those cells to die. So the cells of the heart and the skeletal muscle are most impacted. Now, Tay-Sachs disease is a genetic disorder, and it's about the deterioration of the nerve cells, but a very similar type of issue. The lysosomes are not working properly. So what happens is harmful amounts of the cell membrane components, there's these in the nerve cell, there's these gangliosides, and they end up accumulating in the brain cell because the lysosome doesn't go and digest it and get rid of it. So then when you get that buildup, it eventually, those cells burst and they die. So it is really important that all these organelles function to the best of their ability and function in their role. Here's an example of how it all kind of flows together. Over here, you're going to have your nucleus that's going to be creating stuff. You then have the substances go out the nuclear pores to the endoplasmic reticulum. It will travel along that transport system. Then you'll have the blebbing of the transport vesicle, which will take it to the cyst side of the Golgi apparatus, which will then package it all up. Then it will bleb off again, possibly into a lysosome, depending on what's happening. Often you will have food sources coming in through endocytosis. That food source will connect with the lysosome, whether that was an enzyme that was made or not. That enzyme will help digest it. And that's just kind of the flow of what all happens in the cell. It truly is amazing. So here is just another way to kind of show that exact same process. You have your lysosome, it connects with your food vacuole, which then has the digestive juices. It breaks all that food down to nutrients. It then will spit out those nutrients into the cell as it is needed. Now, we've mentioned those ribosomes as we talked about rough ER. Ribosomes can also be free within that cytoplasm. It doesn't have to be attached to the ER. Ribosomes contain rRNA. There's actually it's a double unit. There's a large part, a large subunit, and then a small subunit. And it is the site of protein synthesis, and it is very important for those cells. Now, there are some sites that have quite a lot of proteins to make, such as the pancreas, and those have lots of ribosomes just littered all over both the ER and the cytoplasm. They are a very important part of our body. Protein is just so essential for all the different amazing functions that the body does. Now, we're going to take a look at the mitochondria, and mitochondria are a specialized cell or organelle inside the cell, and their whole purpose is they burn glucose to produce ATP, and that is a big part of how the cell gets energy so that it can do the things that it needs to do. One of the formulas that you do need to know is this formula right here, which is called cellular respiration. Now, this is not the breathe in and out, the inhale, exhale that we think of. Cellular respiration is when the cell uses glucose, which is this right here, glucose, and it takes oxygen, and within this mitochondria, it is going to burn that, and it releases carbon dioxide, which then will be removed from the cell, goes into our bloodstream, and we do breathe out. It will produce water, but it will also give the cell energy so that it can do its function. It is considered the powerhouse of the cell. It's all made up from these cristae. That's the structure, those cristae. Now, there is also the cytoskeleton, and there are three main parts of the cytoskeleton. There's microtubules, microfilaments, and intermediate filaments. And this is what gives the cell the shape that it needs to, all three of those. Microfilaments are super thin, and they're protein fibers. They usually come in bundles. Similar in composition to the protein in muscle. So it's kind of what allows for that muscle contraction. And you get pinching off of these during cell division. Another type of the cytoskeleton is the microtubules. The microtubules are typically found in the cytoplasm, but microtubules will also make up cilia and flagellum and those centrioles that are so important in cell division. You will hear some examples. These are the microvilli in your small intestine, and you can see these microfilaments, giving it that structure that it needs, just supporting it a little. Centrioles are small cylinders. They look kind of like a pasta almost. And in most cells, they are a 9+0 arrangement. What that means is you have these 9 triplets. See how there's 3 microtubules, and you have 9 triplets, 1, 2, 3, 4, 5, 6, 7, 8, 9, and then no microtubules in the middle. So these centrioles are 9+2. There's other centrioles around that are 9+. These centrioles are 9+0, but there are other centrioles 9+2, that type of thing. The microtubules are this central structural support of both cilia and flagellum. So they are very important for, for example, sperm that moves by a flagella, that's the microtubule that's giving that flagella the motion that it is needing. Cilia often are in bacteria. They're typically large numbers on the cell, but they also have that cytoskeleton, that support system in them, so that they have the shape and keep the shape that they can. Here is an example. Here is some sperm swimming. This is a undulatory movement. That's that snake-like movement, that up and down that allows it to move forward. Cilia are a lot shorter than flagellum. They still move back and forth. For example, they'll allow bacteria to move through a substrate. They just kind of whip back and forth, that type of thing, to allow, and sometimes they are in, it's like seeing the wave kind of thing. So here's the Coles Notes version. This is the big thoughts. So a cell is a living unit greater than the sum of its parts. So the cell has a bunch of different parts to it, and they are all absolutely critical for the cell to do what it needs to do. While the cell has lots of structures, everything has to work together. As soon as something doesn't work, the whole system can fall apart and the cell can die, as we saw when we talked about Tay-Sachs and Pompe disease. The enzyme of the lysosomes and the proteins of the cytoskeletons, that is all synthesized in the ribosomes. The information for the proteins come from the genetic messages sent by the DNA. All of these processes require energy, and that's why that production of ATP and use of ATP in the mitochondria is just so important. Well, hey, thanks for watching, you guys. Whew, that was a long one. That's a lot to learn and to know. I'm going to try and work on making those next ones shorter. But thanks for watching. Hope you guys enjoyed it, and I hope this is helping your learning. Bye-bye.